All right, it says that I am live. I hope that's true. If so, good morning, everyone. My name is Greg Cox, and I'm here to introduce our first speaker. It's my pleasure to introduce Dr. Brian Trainer. He's an MD and a PhD, so he beats me by 1 degree, certainly. He's a neurologist and senior investigator at the National Institute on Aging at the NIH, and he's also an adjunct faculty member at Johns Hopkins University. He's best known for his work aimed at understanding the genetic etiology of ALS and frontotemporal dementia. He led the international consortium that identified the repeat expansion in the C9orf72 gene that underlies the largest proportion of familial ALS patients. He has over 200 publications in professional journals, so very well respected. These journals include the journal Neuron, New England Journal of Medicine, Nature Neuroscience, and many others. He's very well published. He's received numerous awards for his work, including the NIH Directors Award, the Derek Denny Brown Award, the Sheila Essey Award for ALS research, and the Potemkin Prize for research in PICS, Alzheimer's, and related disorders. He received his medical degree from University College Dublin Medical School in 1993, so international flavor there. He got a medical doctorate from University College in Dublin in 2000, master's in medical science from Harvard University and MIT in 2004, and his PhD from University College Dublin in 2012. He's been a member of the Royal College of Physicians of Ireland since 1996. So before moving to the NIH in 2005, he completed a neurology residency and an ALS neuromuscular fellowship at Mass General Hospital and Brigham and Women's Hospital in Boston. So that's his local New England connection. Yeah. Thank goodness. And he's an instructor and staff neurologist at Harvard Medical School and Mass General Hospital. So with all of that introduction, I'm incredibly pleased to have Brian here to talk about the genetic discoveries that he's making and how that applies to ALS patients for diagnosis, as well as potential therapeutic treatments in the future. So with that, I'd like to welcome Dr. Brian Trainer.
Great. Thank you, Craig. That's really a lovely introduction. Can everybody hear me and see the slide? Yeah. If Amber could let me know that it's there. Yeah. All right. Well, I'm going to assume that everybody can.
You're good.
Yeah, I'm good. Okay. Because I'd be worried if I was off, taking a stroll on my own. That would not be very good. And I also, given that I lived in Boston for 7 years and so forth. I'm just wondering whether it's already been snowing there in New Hampshire and in Maine, because I know you guys get colder a lot quicker than we do down here in DC. But anyway, I want to say thank you again to Craig for that lovely introduction, and of course for the invitation to present to you this morning. I'm going to talk for about 40 to 45 minutes. I am Irish, so I tend to babble on, and I rely on Craig and Amber to actually stop me if I keep going too long. But then we'll leave some time at the end for some questions. Now, what I've been asked to talk about this morning is basically gene discovery and therapy. And I thought that what we should call this is the yellow brick road. Because, I think that's quite an apt analogy. I've got this nice picture here that you're going to see a couple of times throughout my presentation. And we'll go over it because it really is a bit of a long, arduous journey. And I really want to sort of make that point to everybody. But anyway, we'll talk more about that anon. So I'll start off with some of my disclosures. These are the various people who've actually supported us during the years in terms of funding this very expensive work and looking for new genes. You'll notice that it includes the ALS Association, and I'm quite pleased to say that in collaboration with John Landers and Jonathan Glass at Emory, the ALS Association is still supporting us. So we're very thankful for that. Okay, so I also think that it's particularly apt to be talking about ALS this year because it's about the 150th anniversary of the discovery and first description of ALS by this man here, Jean-Marie Charcot. he was working at the Salpêtrière Hospital in Paris, France, where he first described this as a condition. Remember, in those days, neurology, and indeed until really quite recently, last 30 years, neurology has been a very descriptive, observational type of science, where we actually collect patients together that look the same and publish them as case series. And in fact, in France, ALS is still known as Charcot's malady. Now, just to very quickly go over this, ALS is one of those unique conditions that causes both upper and motor neurons to degenerate. That's actually quite unusual within the field of neurology. There's literally only a handful of conditions that will do this, and ALS, unfortunately, is at the very top of that. And as I mentioned earlier, this condition is described by Jean-Marie Charcot and is now still known in France as Charcot's malady. But in America, it's more commonly known as Lou Gehrig's disease, after the baseball player who developed developed ALS he was a Yankee baseball player. But he, of course, is not the only famous person to have died of this condition. Now, one of the reasons why I put this up here, this slide up here showing the various famous people who have died, is that there is a perception, you could almost call it a misperception, that ALS is a rare disease. And I would argue that actually it's a lot more common than we actually think it is. So just to say that Mao Zedong is alleged by one of his physicians to have had ALS towards the end of his life. Normally, if I was in an audience and I was in front of you, I would ask everybody to put up their arms and their hands if they recognized who this chap here is, David Niven. And usually what you will find is the people, if you look around at who's got their hands up, they tend to be the older people, because nobody young today knows who David Niven was. But let me just educate you on this. David Niven, it was essentially the George Clooney of his time. He was a British actor who lived in Hollywood, very famous roles. He actually played 007 in one of the movies, and was really one of these debonair characters, and really good movie actor. He's very close to the heart of most ALS neurologists, because David Niven actually gave the money to start the very first, in his will, the very first ALS association, which was the British ALS Association. And in fact, that particular society still works and operates out of the David Niven House. Okay, excuse me. The gentleman on the right-hand side of your slide is Henry Wallace. And actually, he was a vice president of the United States. He missed out on becoming the president of the United States by 81 days. And that was because he was replaced on the ticket by FDR. He wanted somebody who was not as liberal as Henry Wallace was, and he picked Harry S. Truman instead. So he missed out on actually becoming president by about 81 days, but then ultimately went on to develop and die from ALS. And I'd like to put this point forward, that you could imagine how much funding, additional funding, there would be for ALS if a US president had actually died of ALS. Kind of an interesting thought experiment there. But as I say, I put this up to show that really ALS is not as rare a condition as we actually generally think of it. And it's kind of interesting to look at these interesting faces and to discuss this. But from a scientific perspective, we can also put some numbers on this. And this is an interesting piece of work that was done by a doctor who was working in my lab by the name of Carissa Arthur. She now is working at Hopkins. What she did was she calculated the number of cases of ALS in the world today, and then sort of tried to project out to 2040 how many new cases of ALS would exist in the population 20 years from now. And it turns out that there's going to be nearly 400,000 cases of ALS worldwide, which you can imagine, that's not a small number. That's quite a large number of cases. And it does also, interestingly enough, represent an increase of about 69% over today's number. And the reason for that is not because ALS is suddenly becoming more and more incident. It's actually because the world global population is aging. There's this bolus of young people, people who are young today, but in 20 years, they're going to be entering into middle age and middle life. And of course, that's the time period when you're more likely to develop ALS. So I think that this is really good point of information to get across to those who run healthcare services, because they're going to have to prepare for this bolus of patients that are coming down the road. It's also, I also really emphasize this whenever I talk to drug companies, because I try to get across the point to drug companies that there are a lot of patients out there that they could potentially sell any drug that they develop, and therefore potentially could make money from, which of course, when you're talking to big pharma, that's really what they're considering. The other point that is worth considering here is that those numbers are based on the median survival of ALS of approximately 3 years, right? So unfortunately, ALS is associated with a rapid progression, and most patients, about 50% of patients, will die within the first 3 years. And the point that I generally make to pharmaceutical companies is that imagine if you develop a drug that prolongs survival by 1 year. You have now increased by 1/3 the number of patients that you can keep selling your drug to. And it's usually at that time that you actually see the dollar signs light up in their eyes, and they kind of realize, wait a minute, there might actually be something that we can do here, and it might be beneficial to everybody, all sides, patients and stakeholders as well. Okay. Okay, so that was my initial preamble, but I just want to give you a quick summary of what I'm going to talk about this morning. I've talked already about how important ALS is to the community, but I also want to spend some time in the next set of slides talking about why we spend all of this money to find genes. Why is it an important endeavor to do? I also want to talk about, given all the efforts that have gone into this, where do we actually stand today in terms of our knowledge of ALS genetics? I'm going to talk a little bit about the discovery of C9orf72, not for any other reason than it was kind of my little baby and what my lab is known for, but more importantly, because it's such a common cause of ALS. And I actually think that, you know, potentially within the next few years, with the advent of antisense oligo treatment, might actually be something that we can do something about and alter the disease progression in a meaningful way. I'm going to talk about large-scale genomic studies and why they have to be large-scale and why they're important to keep going with them. And then I'm going to talk at the end about, you know, can we actually treat ALS? And more importantly, from my perspective, how can we leverage what we know about genetics to help us treat patients with this disease in what's it's a bit of a buzzword at the moment, but it is actually a true buzzword. It's a meaningful buzzword. It's called precision medicine. Okay, so let's get on. So why do we want to find genes? Why do we want to spend millions and millions and millions of money from the ALS Association, from taxpayers and so forth, to actually find these genes? Well, I think that you can divide it really more or less into two different categories. On the one side, you've got the clinical reasons why you want to actually be able to find genes, and on the other side, you have the scientific. And that divide between the two, it's a little bit artificial because they do feed into each other. It's a bit of a circle rather than an actual divide. But for this purpose here, to actually think about it, it's good to sort of tease them apart. So from the clinical side of things, I think it's pretty obvious to say that if you actually know what gene is causing, what genes could potentially cause a disease, this doesn't just apply to ALS, it applies to all diseases. But if you know the gene that causes a disease and you can actually search for that gene, when you see a patient in front of you, it can actually help you establish the diagnosis, and particularly help you establish the diagnosis perhaps a little bit earlier than you would have otherwise. Now, to be fair, in most cases, I won't say this is all cases of ALS, but in most cases of ALS, that you tend to know the diagnosis pretty quickly, because as I had mentioned earlier, there's only a handful of different types of diseases that can affect both upper and lower motion neurons. However, there are a percentage of cases, unfortunately, where it's not always clear what the underlying diagnosis is. And yet, if you actually are able, if you know the gene and you're able to sequence for it and you find that gene, it helps you to establish that diagnosis at a much earlier stage. One thing that I'm particularly excited about in the future is this idea of predictive diagnosis. So this is a concept maybe new to some of you in the audience, but I think it's something that's really going to change how we practice medicine. This is the idea that we don't wait for patients to develop symptoms. We actually predict not only that a set of patients is going to develop disease, but also when. So why would you want to do that? Well, you want to do that because basically it allows you to start treatment at an earlier stage. I think we can all agree that no matter how good a treatment is, it is not going to work on a dead neuron. There's no drug that it will work on a dead neuron. So you really want to start your treatment as early as possible so that there's as big a pool of living neurons. They may be sick, but they're not yet dead. Bigger pool as possible that your drug can actually work on. And so this idea that we can predict based on genetics and maybe family history and some other clinical aspects of disease really starts to come to the fore. And I think that really we're going to, when medical students today come to retire in 30 to 40 years, how they practice medicine is going to be completely different from how we do it today. This is just a paper. I'm not going to bore you too much with the details, but it's an early attempt to apply that predictive diagnostic algorithm to Parkinson's disease, which of course is another neurodegenerative disease. And they actually already have really, really good results. If they're up to 93% ability to actually predict who's going to develop Parkinson's before they actually get sick. Of course, genetic counseling is really important. Knowing the gene is really important for genetic counseling. One of the most common questions I get asked by my patients in the clinic is my family going to get this disease? Are my sons and daughters at risk for this? And knowing the gene, of course, helps us to quantify that risk and indeed screen those individuals to see if they carry the gene and determine whether they might be at increased risk in the future. So that's the clinical side of things. I think from the scientific side of things, it really, and this of course speaks very much to what Craig Cox does, it is really important to know what the gene is in order to actually model the disease. It is almost, I would argue, impossible to model a disease if you do not know the gene. It is the necessary starting point, because how are you going to make a mouse if you don't actually know the gene and you can't inject in that gene and overexpress it within the mouse? That's really quite difficult to do. It is possible, but it's much, much easier if you actually know the underlying gene. It also helps us understand the pathogenesis, and that's something I'm going to talk a little bit more about in the next few slides. And then ultimately, and this is what I particularly like, and is the reason why I got into genetics, to be fair, is because it provides therapeutic targets that we can go after and we can actually try and treat. Okay, but here comes this metaphor that's sort of over sort of an umbrella over this entire presentation, and that is that really this is a yellow brick road. And the point that I'm trying to make here is that the discovery of the genes is not the end of the story. In fact, it is only the beginning of the story. And it's really a long road from the discovery of a gene all the way to first-in-human clinical trials. Now, sometimes you get lucky, and we'll talk about a few instances of that happening later on. But generally speaking, it is a 10-year, 15-year odyssey down a yellow brick road. And we used to say that it cost $1 billion to do it. And actually, I think that that's quite cheap. I think it's nowadays, it's closer to 2 billion. And even investing that 2 billion, the likelihood of success is quite low. And this is a huge problem that faces the pharmaceutical and, of course, the academic side of things as well. And we're always struggling and always trying to come up with innovative ways to decrease that amount of cost and to decrease that time span as well. Okay, so where do we currently stand in terms of ALS? Well, I think one easy way to get that across, to get that idea across, is to show this timeline of ALS genetics. So the x-axis is really just the time. And we start back here. I don't know if you guys can see my mouse, but we start back here in 1993 with the discovery of the first gene that causes familial ALS, which is SOD1. It was found by Bob Brown. It accounts for about 12% of familial cases, so about maybe 1 to 2% of all cases of ALS. And then you'll notice that there's really, and that was a bit of a watershed moment, I will say, for ALS, but then you'll see that there was kind of a long sort of pause, many decades really, that passed, occasional gene being found in here, but nothing very, very notable until we came across TDB43. And then this sort of real sort of explosion over the last few years in terms of the number of genes being discovered. And what that reflects, I believe, and I know, is really advances in the technology that we use for genomic sequencing. So we've gone from this sequencing each gene and each exon individually using Sanger sequencing to what we call next generation sequencing, where we can just blow through the genome at increasingly low costs, and ability to actually go through the 6 billion base pairs to find the underlying cause. And today, I think that it's fair to say that basically, if you look at this graph, the top of the graph represents the familial cases and the bottom represents the sporadic cases. I think it's fair to say that we now know the genetic etiology of about 2/3 of all familial cases and about 10% of the sporadic cases, which is great. I mean, to be fair and to be honest, that is a real sort of movement forward, and I think we should really be quite proud of how we've been able to do this over the last few years. But nonetheless, before we start patting ourselves on the back, I think we should also recognize that this means that there's 1/3 of familial cases that we don't know the genetic etiology, and there's 90% of sporadic cases where we don't know the genetic etiology. Now, to be honest, I think that over time, this 1/3 of the familial cases is going to get smaller and smaller and smaller because we're going to be able to work it out. We're going to be able to find those underlying genetic causes. What really keeps me up at nighttime pondering and thinking about how we'll tackle it is actually on the sporadic side. Now, so that's really a snapshot of where we sit at the present time. A lot of genes out there, some big, some small. But I don't want to give the impression that what we do in terms of finding the genes is just about finding the next gene. Actually, even if the gene is very, very small in terms of the percentage of patients that are affected by it, it still gives us insight, and it provides us with an opportunity to actually try and understand why it is that motor neurons are dying. Now, this is a nice little slide here. It's actually on animation, so we may actually have to go back and redo it. Let me just do that again. And basically what we have here is all of the chromosomes, 1 to 22, with then the sex chromosomes X and Y at the top. And layered on top of that, what we've put in is the different genes that we know cause ALS. And then layered on top of that again is the different types of pathways that they might be involved in. And what you see emerging is basically different pathways emerging, including, for example, proteostasis, the ability of the cells to maintain the proteins, followed by the RNA metabolism, which is really emerging as a key pivotal point in ALS. Also autophagy and cytoskeletal organization. Now, the cytoskeleton is something I want to talk about a little bit later on, but suffice to say that if you think about motor neurons, which are these incredibly long cells, the scaffolding that's required to maintain those really long axons is called the cytoskeleton. And you can imagine how, when you have a cell that's a meter in length, how that is a crucial part to the cell, and why it might be actually a target and a reason why some of these cells are dying. Okay, so let's move on and just pick 2 of these genes and how we actually went around discovering these genes. And the first one I'm going to talk about is C9orf72, and the second one I'm going to talk about is a more recent discovery called KIF5A, which is more on the cytoskeletal side. So in terms of C9orf72, our story really starts back in the sort of, I would say, late 1990s, early 20s, early noughts, with a couple of papers from Vance et al. And Marizzi et al. That were finding an area on the short arm of chromosome 9 as being related to ALS. And when you actually lined all of those different studies up, represented here by orange bars, what you found was that there was this area of about 7 million base pairs on the short arm of chromosome 9 that was tied into ALS and frontotemporal dementia. So 7 million base pairs, that's about the size of the city of London, because there's 7 million people living in London. Not a huge area in genetics terms, about 100 genes in there, not a small area either. But really our involvement in the hunt for this particular underlying gene, because it's good to know, I should preface this by saying that it's good to know where a gene lies, but the real meat lies in knowing what the actual mutation is, what's the underlying mutation. And we get excited when we can localize it down to a region, because it means we can focus all of our efforts on that region. But nonetheless, what we really want to know is the underlying causative mutation. So our own involvement in the hunt for C9 really began with a genome-wide association study of ALS in Finland. Now, it turns out that Finland is a really good place to study ALS for two reasons. Number one, they have one of the highest rates of ALS anywhere in the world, for reasons that will become apparent. And the second is because they are a relatively conserved population, because they've gone through not one, but two population bottlenecks over the last couple of thousand years. And it means that the population is quite conserved. I have, as you can imagine, many Finnish friends, because I work quite closely with the Finnish over the years, over the last decade and a half. And as I like to joke with them, it's really bloody cold in Helsinki, and nobody wants to live in Finland, and that's why it's such a conserved population. So really, we use that to our advantage in order to try and find genes. But anyway, suffice to say that with our genome-wide association study of ALS in Finland, we were able to narrow that locus from 7 million base pairs down to just 100,000 base pairs. Now, 100,000 base pairs in genetics terms is nothing. It is the blink of an eye. It is a street in London, and it only had 3 genes or 3 houses living on that particular street. And I know when I actually saw this particular result, I actually made my poor grad student, Jennifer Shimmick, stay in the lab for 2 weeks solid without going home, because I thought that we really would have the gene that underlay this very quickly. When I rang her to say she had moved on to medical school, and when I rang her 4 years later to say we had finally found it, she reminded me, of course, of the fact that I had made her stay in the lab for 2 weeks solid without going home, and that it only took us 4 years to actually find it instead of 2 weeks. But anyway, that's another story. And basically what we realized from an early stage, from those early efforts, was that this was going to be a more difficult nut to crack. And indeed, there were all these neurogenetics laboratories across the world that were working on this, and similarly to us, not having success in finding the underlying mutation. So what we said is, okay, well, we're the NIH. Let's see if we can do things a little bit differently. So instead of competing with everybody, we said, okay, let's form a consortium and try and work on this together. And you see some of the people that were involved in this here, including my good friend from Helsinki, Pentti Tienari. And we also reached out and formed a collaboration with another consortium led by Rosa Rademakers down at the Mayo Clinic. Now, one of the families that we focused on from a very early stage was collected by this chap here, Hugh Morris, who's this very smart neurologist who at the time was living in Cardiff. And he had identified this family living in a village south of Cardiff. And he spent a decade going down and collecting the various family members and collecting their DNA and looking at their clinical phenotype. And I think that this illustrates a very important point, which is that really it is the clinicians and it is the collection of this good phenotype data that is the starting point for genetics. And sometimes, you know, when we get this stuff into the laboratory and we see it in tubes, we forget that there was a lot of work that went into collecting this, and we shouldn't really forget that at all. So we tried a whole variety of different techniques, none of which was showing anything, any mutation underlying it. Now that was not wasted effort, because really it told us that it was going to be one of two different types of mutations. It was either going to be a complete inversion of the region, or there was going to be an insert of a new piece of DNA into the region, because any other type of mutation would have shown up with what we had already done in terms of the various analysis. And basically what ended up giving it to us was we did some really, really deep sequencing of the region. And in the end, this is a sort of a snapshot, a screenshot of that day when I was sitting in front of the computer and actually finally figured out what was going on here after, I say, 4 years and a lot of hard work. And basically what you see here is deep sequencing. Each of those bars, gray bars there, represents an actual read. We had about 400x sequencing coverage across the chromosome 9. And what we see is this sort of going from 400x down to 2x and then back up to 400x. And that's a pattern that we know is associated with a problem, a structural problem with the underlying DNA. And that was really what gave us the clue, because then I realized that that actually sat in a known repeat made up of GGGGCC right at the start of this gene called C9orf72. And that was really sort of the clue that we needed to understand what was going on here. And it turned out that what it was this insert of this massive repeat expansion, these 6 base pair motif GGGGCC stuck into a particular gene right at the start of C9orf72. And I love the fact that we used this next generation sequencing, which was very cutting edge. And then we went back to a very, very old technology called FISH to actually prove that it's there. And actually, you can just see in this insert here, I don't know if you can see my mouse, you can actually just see chromosome 9 lighting up there, proving with this very old technology that actually that repeat expansion is sitting there on the C9orf72 short arm. This gave rise to back-to-back publications in the journal Neuron in 2011, back-to-back with Rosa Rademakers. And what's particularly exciting about this is that it's such a common cause of disease. It accounts for about 40% of familial ALS and about 8% of sporadic cases within European ancestry populations. And interestingly enough, a similar percentage of frontotemporal dementia. So we're basically taking two very different, clinically different diseases, ALS and frontotemporal dementia, and sticking them together, marrying them together, and showing that they truly were related to each other. Of course, finding genes is really fantastic, and you know, it's what we live for. But every so often, we can actually do a little bit more than that. We can actually use genetics to understand a little bit more of what's going on. So we tried to do that, and actually one of the interesting things was we were able to look at the haplotype, which is the surrounding structure and arrangement and pattern of SNPs around this particular locus. And what we found was that every single patient, no matter where they were, even that sort of green line very much at the top there, which was from Japan, the only Japanese patient at the time that we found carrying the repeat expansion, they all carried this haplotype that we had discovered in Finland when we were doing our genome-wide association study. So what we think happened, of course, there's no way, I've asked the ALS Association for money to build a time machine to go back in time. Funnily enough, they said no. But what we think happened was that this event happened one occasion up in Scandinavia, in Finland. Of course, the question is, well, it's now everywhere in the world, or at least in the European ancestry populations. How did it get around the rest of the world? And it turns out that we think that it was the Vikings on their summer holidays that actually brought it everywhere else around the world. And in fact, if you look at where the Vikings were, they conquered during the Middle Ages, it actually corresponds quite nicely indeed to where we are now today in modern world, finding high incidences of the C9orf72 repeat expansion. So I love this sort of coalescence between history and epidemiology and modern science. I think it's kind of cool from that perspective. So let's see, how much time do I have left? I have about 10 minutes left. Okay, I think I can get through this fairly quickly. So one of the, I do want to very briefly talk about, so C9 OR72 happened about a decade ago. What about more recently? Well, I think that what we are doing is that we are still going forward with large scale studies. And this is a genome-wide association study that we did on ALS, published about 2 years ago. And you'll notice one thing, what I really wanted to tell you and show you this slide, the reason why, is because look at the numbers. 20,000 ALS cases and 60,000 controls. That is a massive number. And actually, I will say that if you were to look at Alzheimer's disease, or if you were to look at genome-wide association studies that have been done in Parkinson's disease, they dwarf this. So even 20,000 cases and 60,000 controls is considered moderately large, not even massive in genomics terms. And what this tells us is that genomics is a numbers game, because you need these massive numbers in order to find these genes to actually tease it apart. And that's actually, and I know that I'm talking to the audience today, but if you have the opportunity to give DNA samples, this is what you're giving. It is ultimately yours on its own, may not actually give us anything, but when you combine it with the other 20,000 or 40,000 cases, it really, really does start to make a difference. And it's only through efforts like that, those large numbers. What we did find was this new gene here called KIF5A on chromosome 12. And it turns out that John Landers, also in New England, working at the University of Massachusetts, he was coming at this in a different way. He was doing exome sequencing in familial cases, and he had actually found the same gene. So we actually had this sort of X and Y intersecting where we were both saying this is KIF5A. So John and myself decided that we would work together and publish together. And it turns out, and I alluded to this earlier, that what this is it is one of those proteins. KIF5A encodes a protein called kinesin. And kinesin is one of these cool proteins that's actually very active. It actually walks down the axons and transports the cargo behind it, dragging it behind it. And you see this cool illustration here. That's an axon with the protein, the kinesin protein, walking along, dragging that vesicle containing the cargo behind it. And what is even more interesting about this is actually KIF5A was known to the neurological field as a cause of another type of neurological disease called Charcot-Marie-Tooth or hereditary spastic paraparesis. Excuse me. Excuse me. Different type of motion neuron, but what we were finding was that the mutations in the hereditary spastic paraparesis, they were actually in this part. I don't know if you can see my mouse, but basically in the stalk, whereas the mutations that were causing ALS were over here in the bit that was binding to the actual cargo. And it's a notion that we're seeing time and time again in neurology, not just in ALS, but in neurology in general, that the same genes are cropping up again and again. And sometimes in one portion of the gene, you'll see it associated with one type of disease, and then in another part of the gene, you'll see it associated with another. And that's kind of an interesting idea. The same characters and culprits keep cropping up again and again. So again, this gave rise to a nice paper in Neuron. The key point here, the one that I'm hammering home, is how many samples we used for this. 125,000 samples. So this is a numbers game, and it's only through help from patients that we can really get to it. And now I'm going to end very briefly. I think I have 5 minutes left. Where we're talking about can we use and leverage what we know about genetics to treat diseases and treat ALS. So remember, I alluded to this idea of the yellow brick road, that it's really finding the genes is the starting point only, and that it's a long road to actually get to a therapy into humans. That being said, and that's a true thing, that being said, every so often you do actually get lucky. And this is a good example of this. I think this is a set of genes that were found in a type of ALS called Brown BVVL, Brown Violetto Van Leer syndrome, which is a very, very rare, very early onset form of ALS associated with deafness and with blindness. But what was cool about this was that they were finding multiple genes, but each of those genes were within the riboflavin pathway. So we actually had 2 different proteins within the same pathway, the riboflavin pathway, that were being affected. And this gave rise to the idea that maybe if you treat these patients with high dose vitamin B2, sorry, I always forget which side of the Atlantic I'm on, vitamin or vitamin B2, you might actually be effective. And Henry Holden, who was instrumental in finding these cases in Queen Square, he's done this, and he's shown quite nicely that it actually slows down and some cases reverses the effect of ALS. So that's very exciting thing. So that actually predates any of the antisense oligo trials that have been done. I think that this is really kind of cool finding. But that only happens every so often. So I think that really, we have to step back and think, well, okay, we can't rely on pure serendipity. How can we actually leverage the genetics? And I think that we should not just think of genetics in terms of giving us just a new target. I think that there's all sorts of different areas where the genetics comes in, and that's it, understanding what's going wrong in the cell. How can we use it to classify patients and to sub-classify patients into different types? Can we use it to establish a diagnosis in an earlier stage, which allows us to treat the patients at an earlier stage, and indeed to come up with and gauge what their prognosis might be, the patient's prognosis might be, in addition to all of the pathology. Now, I'm just going to end really with this slide here, which is really kind of thinking about what is it that really excites me when I see my patients these days in terms of what clinical trials out there are really good. And I think the ones that have just been published in New England Journal talking about the antisense oligos against SOD1 are really exciting. Now, remember that only works for patients who carry SOD1 mutations. I cannot emphasize that. I get asked this a lot by patients, oh, can I take it? Well, do you have a SOD1 mutation? No, I don't. Well, then you can't really take it because it's not geared for your type of disease. So this only works in those 12% of cases that actually carry SOD1. 2% of cases that carry SOD1, 12% of familial. The other one that's going to be really exciting, and I just cannot wait to see the result of this, is the antisense oligos targeting the C9orf72 repeat expansion. I think that the antiretroviral therapy, that there's a trial of that going on at the NIH, led by Avi Nath, who is a really smart neuroimmunologist. And it's based on this notion that really there were a couple of cases of ALS that occurred in patients with AIDS, with HIV. And when they were treated with anti-AIDS, anti-HIV, antiretroviral therapy, they got better. So Dr. Nath has taken that idea and is applying it more generally to ALS patients. So I think that's very exciting. And then there are a couple of other antisense oligotherapies, including Neil Schneider at Columbia, who's working on antisense oligos targeting FUS mutations. So in summary, what we've talked about today very quickly, and I apologize if I've gone too quickly, but hopefully you can ask me questions if there's anything that was not clear, is that the number of ALS cases is increasing over time. It's not necessarily a rare disease. We know the genes for about 2/3 of familial ALS cases and about 1 in 10 sporadic cases. Most of those are actually turned out to be C9orf72. KIF5A is a cause of familial and sporadic ALS, and the key point I keep hammering home there is it's a numbers game. Look at the massive size of the cohort that we needed to find that, and that really, if you think about it, and this is my personal take, and that people can disagree with me if they like, but my personal take on it is that the future lies in gene therapy and precision-based medicine. Of course, I get to stand up here and wax lyrical about all of this, but actually, this is the sum work and total of a lot of people. If you look at our last paper, it had over 300 authors. So I think that gives you an idea of how many we're talking about. And I'm happy to stop here now and take any questions that you might have.
Okay, thank you very much. Dr.
Please,
Chandler.
Amber, don't tell me that none of that was recorded or nobody heard and I have to go back and give that all over again.
Oh, unfortunately for us, it was all recorded.
Good. Okay, good.
So we do have some questions. I'm going to start. I have a question from Roger. And his question is, if genetic testing was done 4 years ago, would it be worth doing it again now to determine if a gene can be identified in an ALS patient?
You mean 4 years ago or 40 years ago?
4.
4 years. 4 years ago.
Mm-hmm.
Yes, I think so. But actually, this brings up an interesting aspect, is that it depends on how that genetic testing was done 4 years ago. Because remember, I was talking about next generation sequencing, where we actually sequence the entire exome or the entire genome. It may be that the genetic testing does not have to be redone, but rather you have to go back into that data that is sitting on somebody's hard drive and look and see if there's anything new there. So it depends on how it was done 4 years ago. If it was done by the traditional old-fashioned Sanger sequencing, then yes, it has to be redone from scratch. But if it actually exists as data and the entire genome and the entire exome has been done, then it's actually just a matter of searching through that data. And there are now increasingly sophisticated tools that allow us to do that quite quickly.
Okay, thank you. And so we have a question from Mark. And his question is, did I hear that the MN cells are over 1 meter in length?
Yeah, I mean, if you think about it, I mean, I'm not that tall, but if you're over 6 foot, your motion neuron starts in your cortex in the top of your head, and it goes down, at least the ones that are controlling your legs, they go down to the base of your spine. And if you put your finger on your head and put your finger on your lower part of your spine, that's about 1 meter, right? If you're over 6 foot. So and bear in mind that if you had the ability, you can't do this, but if you had the ability to take out a motor neuron, it's 1 meter in size on the table, but it's so thin that you can't actually see it with the human eye. You actually have to get a microscope. So it's 1 meter long, and it's so thin that you cannot see it without a microscope. It just kind of boggles the mind, but it also brings across this point about how super, super, super specialized motor neurons are.
Okay.
Which might account for why they're selectively vulnerable.
Yeah.
Sorry, go ahead.
That's okay. Dr. Cox, do you want to ask your question?
Brian, can you hear me?
Yes, indeed.
Okay, good. Given that you're generating larger and larger sample sizes for these patient numbers, will you be able to then start to look at gene by gene interactions or gene by environment interactions across these ALS patients? Yeah,
and I think that you bring up a really good point. So Andy Singleton and myself actually wrote a review article for Neuron a decade ago, and I had a cause to go back and look at it more recently. And basically, in that review, we asked 3 questions, which was, number 1, do gene environment interactions exist? And the answer is yes, of course they do. I mean, all you have to do is look at lungs and smoking to know that they exist. Should we be looking for them? And of course, the answer to that is yes, because they're really important, because if we can reduce that environmental risk factor, we could potentially decrease the number of cases of ALS and prevent them ever from happening in the first case. So again, the notion of smoking and so forth, and indeed smoking has been implicated in ALS as well. And the third question was, well, can you actually find gene environment interactions? And the answer at that stage 10 years ago was no, you can't. So they exist, we should be looking for them, but there's no point in looking for them because we don't actually have the tools. And now, 10 years later, I actually think that that answer has changed, that dynamic has changed, and we are actually in a position whereby we have so much genetic data and actually so much environmental data as well, the exposome, that we can actually start to look at this in a hypothesis-free, data-driven manner, which is something that I didn't emphasize so much today, but it's this idea that we try and move away, in our lab at least, we try and move away from generating hypothesis, and what we're really trying to do is we're trying to look at the data and let the data tell us where we should be going. So a roundabout answer, but the answer is yes.
That's great. Thank you.
Okay, we have a question from David. Do you recommend genetic testing on all ALS patients? And are there any studies that look at the genetics of fast versus slow progressors, bulbar versus limb onset?
Yeah. The answer is yes and yes. Okay. So my feeling about this, now bear in mind that I put up my disclosures and that I hold a patent on the testing for C9orf72. So I want to make that clear, although I will say that I have received $0 from this ever since we actually issued the patent. So I say it, but I'm not getting really any money for this. But my feeling about this is that every patient with ALS should be screened for C9orf72. And then if that's negative, then you go on to the second layer where you test the less common causes, and then you go down into the really rare nitty-gritty stuff. And you can debate whether sporadic cases need to be screened for the really rare nitty-gritty stuff. That you can debate that, and that's a very valid debate. But I do think that in the next 5 years, as treatments begin to emerge for these rarer and rarer forms of ALS, that debate will go away, and we will just screen everybody for genetic causes, because we know, even though they're incredibly rare, yes, there's something we can actually do about that. So I think that it's a really important question. It's a question that's evolving over time, and that even in the next 5 years, which is a relatively short period of time, that the answer to that question is going to change dramatically.
Okay, so I have a question from Maria, and I'm not sure if she's asking about B12 or B2, but she says, what did B12 do?
Well, B2, so riboflavin with vitamin B2, which is also called riboflavin, same name, two names for the same thing. In those patients with those mutations and who presented with the Brown-Violetto van Leer syndrome, remember, very, very, very rare. There are 22 cases described in the literature of ALS. They actually stopped progressing, and some of them actually even got better. So it really, really worked in those cases. I am not advocating that every ALS patient starts taking vitamin B2. This is an example of precision guided medicine, where the medicine meets the underlying cause. If you don't have that underlying cause, that precision medicine is not going to work. It really and I apologize for being so assertive about that and being so strident about that, but it is a very, very important point, and I find I often have to say it a couple of times to get that point across.
Okay, thank you. I am going to wait just a few more seconds here. We don't have any questions in the chat or Q&A, so if anybody has a question, we have a few more minutes with both Dr. Cox and Dr. Trainer. So if anyone has any questions for Dr. Cox, I'm sure he'd be happy to answer those. So while I'm waiting, I'm just going to remind everybody that in order to join the next session, which is the gamification, I'm sorry. Yes, is the gamification. You just need to remember to hit the join next button, and there may be a little bit of a delay. Technology is wonderful. And also just to remind everyone to, after gamifications, make sure you go into the expo and talk to our wonderful exhibitors and sponsors, who we'd like to thank as well for supporting this programming.
Remember, I have one more thing I'd like to ask Brian while we're waiting for, it looks like Roger's coming up with a question.
Yeah.
So Brian, how do patients get their DNA samples into these large studies like you were talking about, for these genome-wide studies?
Yeah, and actually that I thank you so much for asking that. You can always contact me. We can arrange for it package to be sent out. We can do a collection via mailer, or oftentimes if you ask your neurologist who you're attending, they often know how to do it locally. Both of those approaches are absolutely fine. Really, you know, it always bothers me when I hear that somebody is an ALS patient and they haven't been offered an opportunity to partake in genetics. I always think that's a missed opportunity, because maybe they have the mutation that explains all of this, right? And if we had only just collected this, we'd be further along. So the other thing to think about is you're not necessarily helping yourself, but you might be helping the next poor person coming down the road with this disease. So you're kind of thinking of this for the greater good and the greater community of ALS patients.
Okay, thank you. We do have another question from Roger. Is there a specific name of the test you would recommend to identify a gene?
No.
Okay. So basically,
No.
talk to your neurologist about getting genetic testing and then having
yes.
it. Okay.
Yes.
Because those gene-specific tests are great, like you said, for C9orf72, but that only finds what we already know about. That doesn't allow us to find anything new. And that's what some of Brian's studies, where he's looking across the entire genome, not guessing what the gene is, he's letting the data tell him what the gene is. And I think those are the studies that are going to give us the next discoveries.
Yeah.
Okay.
Yeah. Very much.
And
thank
Justin is saying, what a great presentation, and thank you very much.
Marshall, my pleasure. Thank you.
And I want to add our thanks as well, Dr. Traynor and Dr. Cox, for participating in our research presentation today. And we will, we're going to end the presentation. So there will be a delay between this presentation and the 11:15 presentation. So a good time to take a quick break and come back and join us for gamification and the expo. So thank you again.
All right. Thank you.
Thank you all.